immunology, virology. Our session chair is running a little bit late, so we're going to go ahead and get started. This is Mansee. She's going to talk to us about Gammaherpesvirus, and I'll just let you take it away.
Okay. Thanks, Nathan. Okay. So, hi guys. Good morning. My name is Desai, and I've been working in the Glaunsinger lab, which is a virology lab here on campus. And the title of my project is Characterizing Mutants of Global mRNA Degradation During Gammaherpesvirus Infection. And so as I go, I'll break this title down, it'll make more sense if it's a mouthful right now. Okay. So starting off broadly, all herpes viruses are double-stranded DNA viruses with large genomes that can encode up to 200 proteins. And the hallmark of this herpes virus is a two-phase life cycle consisting of a lytic and a latent phase. And the lytic phase is when a virus enters the cell and it actively replicates. So it releases infectious particles that can go on to infect other host cells. On the contrary, in the latent phase, the virus does infect the cell, but it kind of hides. It doesn't actively replicate. And so this picture over here is a picture of a herpes virus with this outer lipid envelope, and then inside this structure here is what contains the genome, and this genome is linear double-stranded DNA. So Gammaherpesviruses, why we're specifically studying them. And Gammaherpesviruses are of primary interest to us, to humans, because of the two herpes viruses that are under the Gammaherpes viruses that are human viruses. Specifically, we're looking at Gammaherpesvirus, KSHV, and Epstein-Barr virus, EBV. KSHV is actually the leading cause of cancer in untreated AIDS patients, and right now there's no vaccines or antiviral treatments available to treat this. So an understanding of the lytic and latent life cycles and how they interact would be critical for development and strategies for control and prevention of Gammaherpesviruses driven oncogenic diseases. And so all Gammaherpesviruses are lymphotropic, meaning when a Gammaherpesvirus infects a cell, it has a tendency to infect B cells, which are part of our immune system, and it has a tendency to establish latency of B cells. And so in our lab, we're using MHE68, which is a mirroring Gammaherpesvirus. It's a derived version of Gammaherpesvirus, and MHE68 of mice, infection of mice, provides this ideal small model, small animal model system that can, help us understand the function and biological relevance of Gammaherpesviruses. So the disease progression is similar, and it's closely related to the human Gammaherpesvirus, because that's where we're using it. Okay, so what Gammaherpesviruses do when they enter the host is they alter the host cell's mRNA levels, which eventually are mRNAs, which are eventually translated into proteins for the cell. Gammaherpesviruses, they actually alter gene expression by targeting the mRNAs for degradation. And so what we have here is this Gammaherpesvirus, MHE68 is specifically entering the host cell, and then it encodes for this viral protein called musox, represented by this Pac-Man over here. And what this musox does, it cleaves and targets cytoplasmic mRNAs, which are these white things right here. And so those are cleaved, and then musox is still intact, it's still in the cell. And the process of cleaving the host mRNAs is known as host shutoff, and which basically shuts down the functions of the host cell, because if the mRNAs that eventually go on to make proteins for the cell are not there, then the cellular functions can't be continued and the host essentially shuts down. So the previous slide talks about how the wild type or the normal version of musox works. What we did in our lab was we made a mutation in the DNA that encodes for the musox protein, and we created a virus that contains this mutated musox. And we named this virus MHE68 R443I, which is right here on the right. And R443I is just the name of the mutation, and this is just based on nomenclature rules. And what this mutation in musox does is it renders the mutant musox incapable of targeting including cytoplasmic mRNAs to a certain degree. So if you cannot target and include cytoplasmic mRNAs, like the wild type version over here, then essentially it's defective for host shutoff. And so after making the defective MH368 virus, virus, the next step is to infect mice and how it behaves inside the mouse or in vivo. And so usually you infect the mouse through the intranasal way, and then the virus makes its way down to the lungs, replicates in the lungs, and then it makes its way down to the spleen and establishes latency in the spleen. What we found was that 17 days post-infection, we see that the amount of virus is reduced in the spleen compared to the wild-type virus. And so hence, in vivo in the mouse, the mutant virus is not able to establish latency in the spleen as well as the wild-type virus does. So this speaks to its defectiveness. We also observed that when we infected cells in a petri dish or just in vitro, we found that the defect is dependent on the type of cell that you infect. So for instance, in fibroblast cells, which are cells that make up connective tissue in mammals, there is no defect. So we saw a roughly equal line of the host strep deficient virus and then the wild-type version of the virus that infected fibroblast cells. On the other hand, in dendritic cells, which are part of our immune system, we did see a defect. So there was reduced amount of virus compared to the wild-type version of the virus. And this just goes to show that this host shell deficient virus has cell-type specific rules. And so apart from the R443I mutation that I was just talking about, we later discovered two other independent host shell mutants. So these mutants, musox mutants, are incapable of degrading mRNA to a certain degree. But as of right now, we don't have viruses that contain these new versions of musox. But these two mutations, L132F and NOS2, which are here on the musox protein, they become slightly more important later in the presentation. So for now, back to the R443I mutation I was talking about. So I earlier mentioned that it's defective, but only to a certain degree. And this in vitro experiment here with R443I musox shows evidence of this. So this graph over here is measuring the relative mRNA levels for this gene called PSL3. And what we have here is the control, which doesn't have any musox. The wild type is the normal version of the musox. And then we have the mutated version of the musox. And we basically infect the cells with each of these three types of musox. And we see that in the control, there's 100% of the transcripts, which makes sense because there's no musox. There's going to be no degradation. Wild type, there's about 40%. And with the musox, there's about 70% of the transcripts, which shows that, again, musox is not 100% effective. Otherwise, it would have had equal amounts of mRNA transcripts compared to the control. So these two are not equal, hence it's not 100% effective in R443I. Which leads me to think that is it possible that if we introduce more than one mutation onto musox at the same time, and create a double mutant or a triple mutant, would we see greater range of defectiveness? So that leads to my first question, which would be to one, test the degree of defectiveness of an independent host shell mutants, namely L132F and NLS2, as well as compare the range of defectiveness with double and triple mutants. So we went about doing this by making these plasmid spurs. And the plasmid is essentially a circular piece of DNA that encodes for certain proteins. And so we made these plasmids by one, introducing the musox gene with its appropriate mutation, and two, this green fluorescent protein gene that essentially serves as a marker gene. So any cell that has taken up this plasmid will fluoresce. So it ensures us that the cell has taken the plasmid. And so the plasmids we've made with this method are the R443I single, the L132F single mutation, NLS2 single mutation, double mutation with the L132F and the R443I, and then a triple mutant with all of them. And so the next thing we did was we wanted to assay the protein levels of the musox projects that we've made. And so depending on the thickness of the bands here, that tells you how much of the protein is being expressed. And so we have here the control, which has no expression because it doesn't have any musox. There's a wild type where we do see expression of musox. And then there's a single R443 and the double mutant here. And we see roughly equal expression levels. But the important thing with this is that with the triple mutant here, there is no expression, which means that if it's not being expressed, we can't really test how defective it is. So then for future tests that we did, we had to remove that from our future quantification tests. And so from the Western blots, we move on to another quantification test called that actually looks at the defectiveness of the musox constructs we created. And so this was done by measuring the mRNA levels of cellular genes using this process called qPCR. And before we could actually do the in vitro experiment with the qPCRs, we had to make sure that we were working only with the GFP positive cells, so essentially cells that had taken up the plasmids that we made. And so to ensure that we're working with only those cells that are taking up the plasmid, we do this process called cell sorting. And we take these cells, we introduce the plasmids into the cells, and then plate them over a period of 24 hours. Some cells, fluoresce, some don't. We take all of them, put them through this sorter, and then through the sorter we get our pure population of GFP positive cells, and that's what we do our qPCRs with. And so the actual process of the qPCR. So once we have the GFP positive cells, we would do the qPCR and test the mRNA levels of these three genes here, LDHA, PKDRC, and GAPDH. And then upon infecting the cells with the single alpha-4 the L132F, and the double mutant, we measure their cellular mRNA levels. And this is a direct measure of how many of the mRNA transcripts are being degraded. And so the results of these qPCRs will give us bar graphs that tell you the relative mRNA abundance levels of these three genes. And so what we have here is each of these genes is an average of 3 trials for qPCRs. And so directing your attention over to this first graph here, we have on the x-axis and the y-axis, we have the relative mRNA abundance, and then we have GFP wild type, and then alpha-4 L132F, and then the double mutant here. And what we see is that we made GFP wild type equal to 1, so we can measure the relative defectiveness among the musox mutant constructs. And we see that with the alpha-4 and the L132F, they're roughly equally defective, whereas the L132F is slightly less defective. Moving on to this gene here, same pattern here. These two are roughly equally defective, while this one is less defective. And this third gene here, the difference is less exaggerated, but you see a similar pattern with this alpha-4 and the double one being more defective, whereas L132F is slightly less defective. Okay, and so for future directions, I will want to continue the in vitro experiments to test the range of defectiveness of this third mutant, NLS2 mutant that I mentioned, that I hadn't included in my earlier qPCRs. And so I want to test the defectiveness of that and compare it to the other musox construct we've already tested. And then specifically in the context of the virus, I mentioned the phenotypes of the R443 I've already stated, but I want to make another NO268 virus that contains the other point mutation L132F and basically compare the phenotypes to the that we saw with the R443 virus. And so I'm making these multiple viruses, to give us confidence that the phenotypes we see with the R443 are, directly due to the lack of post-op and not some other variable. And eventually I'm going to go double mutant virus and see if the single mutant viruses, if more mutations translate to higher phenotypic severity or not. And as far as acknowledgements go, I would like to extend a special thanks to my PI, Britt Glaunsinger, my grad student, Emma, as well as Nathan, who helped us put these presentations together, and of course the Rose Hills Foundation for providing the monetary support for